Have you ever wondered why you can't see color at night? No? Well, I'm going to tell you, my pretty, and your little dog too. I also have some other information for you, including chemical names you won't remember and the ridiculously confusing process that happens every millisecond your eyes are open. So let's get started. Let us journey into the retina of the eye to find the lost presentation topic. We pass through the cells of the retina into the innermost layer where two types of cells reside. We zoom magnificently on one. It is the elusive yet actually really plentiful rod cell. First, here are several pictures of the rod cell, so I can look like I did more research than I actually did. The thing at the end is called the outer segment, and it is filled with tiny discs. You might wonder why these matter. It's the pigment inside that matters, and it's the part which allows you to see. The other parts are the inner segment, which contains energy-making mitochondria. Actually, they make ATP, or adenosine triphosphate, which can be used by the cell as energy, but I really don't have time to explain that. There's also a little bulge of nucleus where all the DNA is stored. At the end, there's the synaptic terminal, which is the part that sends messages to the brain after the messages go through an information processing process. Does that make sense? Great. Okay, now to the really important part, besides the bits that keep every cell in your body alive. They don't count. The pigment. The pigment inside the pigment disc is composed of a protein, opsin, linked to a molecule of retinol. This dynamic duo is what lets your eyes cut through the night like a speeding bullet, and together they are known as rhodopsin. Now for the confusing part, how your eyes work. When a photon light strikes a rod, the opsin tenses up and changes shape, causing the retinal molecule to also change shape, which causes the molecule cGMP, cyclic guanosine monophosphate, to break down into 5'GMP. The reduction in cGMP causes the ion channels to close, preventing an influx of positive ions, which hyperpolarizes the cell, which sends messages onto the information processing process. That was the longest time without jokes I've had since I became a narrating voice. Aura isn't being hit by light, it releases glutamate, opens the ion channels, and depolarizes. Sometimes if you see little flashes of color, but everything else is black and white, not only your rods are operating. Cones see color, but I'm not at liberty to explain why. The cones need brighter light, and sometimes enough light reaches them to set them off. Vision using both rods and cones is called mesopic vision. Vision using only rods is called scotopic vision. There is a name for vision using only cones, but I'm not at liberty to tell you that. You might find out without my help, though. If there were only rods, however, your vision would be pretty useless. First, rods can't see color. They only have one type of pigment which either registers light hitting them or no light hitting them. And they also can't do the information processing process on their own. Rods are pretty much useless in bright light, which is why you need cones. With only rods, your eyes would be useless. Here is the juice of that part of my brain which stored all that information. Drink it up. You're welcome.